Saidina Umar Al-Hatta. Their project is entitled Rate of Decomposition, Mixed Species Litter and Single Species Litter. Which one is more beneficial as a source of plants' nutrients? The team leader is Siti Nur Hamiza Al-Ghazi Salfi binti Haji Muhammad Bakar. Please give a warm round of applause to the team from Sekolah Menengah Saidina Umar Al-Hatta.
Your Royal Highnesses, Assalamualaikum warahmatullahi wabarakatuh. Our title is the rate of decomposition between single species leaf litter and mixed species leaf litter. Our objectives are: first, to investigate the different rate of decomposition between single species leaf litter and mixed species leaf litter. Second, to quantify the abundance of the part between single species leaf litter and mixed species leaf litter. Third, to study the relationship between the abundance of water part and the rate of decomposition. Lastly, to indicate the decomposable and the importance of decomposition of leaf litter. Decomposition is the process of breaking down that organism to simpler forms of matter. The decomposers are fungi and bacteria. During decomposition, nutrients are released back to the soil to be used by the plants for healthy growth. Our hypotheses are: first, mixed species leaf litter will decompose faster than single species leaf litter, and second, mixed species leaf litter attract more part than single species leaf litter. Mass loss of leaf litter was used as an indicator to measure the rate of decomposition. The more mass loss, the faster the rate of decomposition.
The leaf litter that we use were first rambutan of the Bdellium Acacium, second guava of Physacium guajava, third acacia or acacia species, and lastly mango or Mangifera species. We did our fieldwork in the farm at Kampong Junjungan. Next, on methodology. The first experiment is average dry mass. Average dry mass was used as average dry mass. Average dry mass was used as the second experiment is mass loss. Mass loss was done to find the rate of decomposition of leaf litter after the sample was placed at the study site for 3 weeks. The third experiment is Tugger funnel is an apparatus used to extract living organisms, particularly arthropods from sample of leaf litter. And the last experiment is CN ratio. CN ratio is carbon to nitrogen ratio. In CN ratio, carbon aging, geothermal digestion, and distillation were used. Both experiments were carried out at Brunei Agricultural Research Center with help from the soil specialist Dr. Dipas.
Now we move on to result. According to our methodology, we placed 5 grams of each leaf litter samples at the study site. However, when we carried out average, the actual gram mass of the leaf litter samples ranged from 4.49 grams to 4.85 grams. This shows that the actual gram mass of the leaf litter samples were actually less than 5 grams. Now we move on to result for percentage of mass loss. For the first 3 weeks, we did a research study on guava and acacia leaf litter between the single species leaf litter. Guava has a higher percentage of mass loss. However, when it is compared with mixed species leaf litter, mixed has the highest percentage of mass loss as shown on the bar graph. For the next 3 weeks, we did a research study on mango and acacia leaf litter between the single species leaf litter. Acacia has a higher percentage of mass loss. However, when it is compared with mixed species leaf litter, mixed has the highest percentage of mass loss as shown on the bar graph. For the last 3 weeks, we did a research study on rambutan and acacia leaf litter between the single species leaf litter. Acacia has a higher percentage of mass loss. However, when it is compared with mixed species leaf litter, mixed has the highest percentage of mass loss as shown on the bar graph. From the overall results shows that mixed litter always has a higher percentage of mass loss than every single species leaf litter samples.
Next, on Tugger funnel, total abundance of arthropods. For the first 3 weeks, we used guava, acacia, and mixed species leaf litter. For another 3 weeks, we used mango, acacia, and mixed species leaf litter. For the last 3 weeks, we used rambutan, acacia, and mixed species leaf litter. From the overall result shows that mixed species repeater has the highest total abundance of arthropods for all 9 weeks.
Next, we move on to the discussion. First, I'm going to discuss on average dry mass. Initially, the repeater we used in our study is 5.0 gram. However, according to our result, the actual dry mass is less than 5.0 gram. This is because the water content in the repeater was removed when the samples were overnight for 3 days. The average dry mass is the actual mass of repeater used each week. Next discussion is on Tagong funnel, where used to find the abundance of arthropods. From our result shows that mixed species repeater has higher abundance of arthropods than single species repeater. This is because, according to Blair 1990, mixed species increase the heterogeneity of the sources and change the abundance of decomposers.
Now I will discuss on percentage mass loss and rate of decomposition. Among the single species repeater, each has different percentage of mass loss, hence different rate of decomposition. Starting from the fastest rate of decomposition, guava, acacia, rambutan, and followed by mango. However, the mixed species repeater has the highest percentage of mass loss compared to every single species. Therefore, the fastest rate of decomposition. Lastly is rate of decomposition and abundance of arthropods. From our result, mixed species repeater has the highest percentage of mass loss, and it also has more abundance of arthropods than the single species repeater. Blair conclude that mixed repeater increases the heterogeneity of resources and attracts more arthropods. Hence, the abundance of arthropods causes the mixed species repeater to have higher rate of decomposition than the single species repeater.
We identified that our research study can be divided into economic, social, and environment. First, on economic value. In Brunei, fertilizers are subsidized by the government. Therefore, if we can cut down the usage of fertilizer, we could reduce the government expenses on fertilizer. It is also economically beneficial to the agriculturists. Next, on social value. It can educate the local people on the importance of decomposition of repeater and to provide information for the farmer and the agriculturists on how to naturally increase nutrients in the soil rather than using fertilizer. Lastly, on environmental value. If we implement the use of decomposition of repeater, we could reduce the open burning of repeater. The repeater can also help to reduce soil erosion in forested area and helps to maintain the soil moisture. Last but not least, repeater can prevent the eutrophication, as the eutrophication is an effect from the overuse of fertilizer.
As for conclusion, we can conclude that mixed species repeater decompose faster than single species repeater, and mixed species repeater attracts more arthropods than single species repeater. Terima kasih. Thank you.
Thank you very much, Sekolah Rendah Sedina Umar Al-Khattab. The last presentation will be from Chung